Hello everyone. Welcome you back. Today we'll discuss about one more cell organelle, a very important one. That is what the Golgi apparatus. So it is also called as what the cells shipping and sorting center. Okay. What is Golgi apparatus? First of all, the Golgi apparatus, it is also known as the Golgi complex, Golgi body, or simply the Golgi. Okay. It is also a very important organelle found in most of the eukaryotic cells, right? Part of the endomembrane system. So yesterday class we discussed in the last class, okay, endoplasmic reticulum, in that we discussed endomembrane systems. So it is also one of the endomembrane system in the cytoplasm. Okay, it packages proteins into membrane bound vesicles inside the cell before the vesicles are sent to their destination. We'll discuss next, okay, the main function and how it is going to carry out the proteins and lipids, okay, what it is mainly synthesized by the endoplasmic reticulum, right? So after leaving the endoplasmic reticulum, many of the transport vesicles travel to the Golgi apparatus, okay, the cell's warehouse and distribution center. What is that? Mainly the transportation, that system, okay. The Golgi apparatus is mainly responsible for receiving, modifying and sorting as well as shipping the products which are mainly synthesized or made by the endoplasmic reticulum, okay, such as the proteins and the lipids. Yeah, it can even perform some manufacturing functions of its own, okay. So here you can see that the main structure of the Golgi apparatus. Structurally, the Golgi consists of mainly the flattened sacs, okay, membranous sacs called the cisternae. Okay, this we can call it as what the cisternae. So how it looks like? It looks like a stack of pita bread. Okay, here in this picture you can see that it is also similar to that of the cisternae of the Golgi apparatus. That's why it is simply called as what the stack of pita bread. Each cisterna, mainly it is separates its internal space from the surrounding cytosol. So it is what the cisterna, how it is it separates its internal the space, okay, internal space from the surrounding the cytosol, okay, in the cell. There are many usually many stacks of cisterna in a cell, okay, especially in those specialized for the secretion like gland cells, okay. Around the Golgi you will often find some of the vesicles, okay. These vesicles are tiny sacs, okay. This you can see that these vesicles, these vesicles, okay, these are many stacks of these cisterna in a cell, especially in those specialized for the secretion like gland cells, okay. Around the Golgi you will often find many vesicles. These tiny sacs, what it will do, it will transport the materials between the Golgi and the cell organelle, between the Golgi apparatus and the other cell organelle which are there in the cells. So the Golgi apparatus, okay, it is having the two main distinct the phases, okay, that define its direction of the transport. So as we can see that okay, here in the cis and the trans Golgi. So cis Golgi, it is almost situated or placed near the endoplasmic reticulum, right? And the trans Golgi, it is situated outside of the endoplasmic reticulum, right? So how it is going to work, we'll see, okay. Based on that, okay, the Golgi apparatus mainly it is having the two phases, cis phase and the trans phase. So in the cis phase, what happens, it is going to receive the receiving side. It is also called as what the receiving side, usually located near the endoplasmic reticulum, okay, vesicles from the endoplasmic reticulum, it fuse, okay, and adding their membrane and the contents. So what happens? So and again, when the things are going to be synthesized in the endoplasmic reticulum, so it is going to receive from the endoplasmic reticulum, and it is going to be processed, and through the Golgi apparatus, okay, transphase, it is going to be what, transported that, or it is going to sorting and transporting what processes are occurred to other the cell organelles in the cell, okay. So this is what about the cis phase. And in case of the trans phase, these are shipping side or transporting side, where new vesicles pinch off, okay. The Golgi apparatus, it mainly serves as a central hub in the cell's endomembrane, the system, okay. Then Golgi apparatus, what are the main functions of that? First thing, the Golgi apparatus, it functions as the cell's packaging and the distribution center. That is how it is going to modify that and sorting, okay, as well as it is going to ship those materials made or synthesized from the endoplasmic reticulum to the final destination, okay, to other the cell organelles. So the second one is, as the product moves through the cis and the trans regions of the Golgi, okay, they undergo a series of modifications. For example, glycoproteins, okay, the glycoproteins in that, so it is going to formed in the endoplasmic reticulum, right, have their carbohydrate components altered in the Golgi complex. So certain sugar monomers are mainly removed and others are added, okay. That is how the modifications mainly occurs, are series of modifications, and creating a great variety of the carbohydrates. The Golgi may also modify membrane phospholipids during this transit or this modification. So it is going to even modify the membrane, what? Membrane phospholipids. So membranes also having some phospholipids, right? Membrane phospholipids, okay, during this transit. So next function is, so in addition to the refining endoplasmic reticulum products, okay, the Golgi apparatus, it manufactures some of its own macromolecules. Clear? How? Many polysaccharides which are secreted by the cells, such as pectins, okay, that is pectins, and incorporated into the cell wall along with the cellulose. Clear? That is what, and also these are what, the pectins and the cellulose are the non-protein products, okay. These are mainly packaged in transport vesicles that bud off from the trans phase, okay, and it is later fused with the plasma membrane for the final the secretion of those, okay, the non-protein products. That is what the cellulose and the and that. Then next is the processing and the manufacturing within the Golgi. It mainly occurs in stages with the different cisternae containing unique enzyme sets for the specific modifications. Clear? And the next function is, so before the materials are leaving the Golgi apparatus, the trans phase, okay, it sorts and labels them for the delivery, okay, like a special tags, for example, like the phosphate groups, okay, like that. So that work like a zip codes to show where each product should go and should reach to other cell organelles and all, right. That is what mainly occurred in by the trans phase of the Golgi apparatus. So some of the small transport vesicles, okay, it form from the trans phase and carry these products. So each vesicle has a surface molecules, okay, that help it find and attach to the correct organelle or the plasma membrane. This ensures that every cellular package it reaches the right destination. Okay, this is what all about today's class. So if you found this video helpful, please like, share and subscribe my channel for more curious biology concepts. This is Savita, igniting curiosity in life science. Thank you. See you in the next video.